My name is Christoph Gerber. Ever since we have developed the AFM, I stayed in that area because I'm very interested in nanomechanics, and have taken nanomechanicals applications into the realm of personalized medical diagnostics, and that is what I'm going to talk about today.
Thank you for inviting me to speak here. It's a lovely place, fantastic university. So what I would like to give you a sort of an impression, the way we have taken AFM technology into the realm of personalized medical diagnostics. And that's how it all started. That's the first AFM, and it has some analogy, at least in the making of with this device, which we all absolutely using so marvelously today. But it's just in the making of it. Just sort of gives you a sort of a hint that for proof of principle, not necessarily you have to build a Ferrari. So it lives now. I have been asked. It lives now at the Science Museum in London. And in an exhibition known as the Making of the Modern World. And I believe the Apollo capsule in the background is in good company. So talking about space, this is what I would like to share with you just at the beginning, and that is the European mission ROSETTA, which actually met its target, that a Churyumov comet, after 6.4 billion kilometers of traveling. And it is now circling actually around the this comet, and you see that here, in about 30 kilometers, and then it will touch down a lander in November. And as we are speaking, actually, they have 11 systems, mostly spectroscopy on board, and they have already found all life molecules already with some of the spectroscopy system. However, it also has this on board. This is MIDAS. It's an imaging dust analysis system, and it basically consists of an AFM. And obviously, this is technology that has been developed 15 years ago. However, after hibernation of this complex sort of bypassing in the space, they switched on the system again, and what came out is this. So they are actually investigating some particles here. But a keen AFM eye actually sees that this carries some multiple tips, so nobody knows actually at the moment what it is. However, it's working, and it's working for another half a year or a year, and certainly they will come up with some beautiful results, no doubt. So what I'm concentrating is actually today to share that with you, as we just heard already in this conference, the progress of AFM in biology is quite substantial in the last 2 years, and we heard that in talks yesterday and today. So I will just concentrate on a few things of it and show that AFM technology indeed is capable of doing in principle that it entered the clinic in some ways, and I will show that here. Here it is a diagnostic tool used in force distance spectroscopy, where diagnosis from patients' tissues, and there you can see that the diagnosis for breast cancer, this is basically a malignant cell, and the diagnosis for breast cancer at present relies on histological staining. And the breast cancer tissues exhibit heterogeneous nanomechanical properties, and that basically allows to study the structure of the extracellular matrix by creating distinct fingerprint. And you can see this here. This is normal tissue, this is benign tumor, and this is invasive cancer. And you do that by measuring thousands of measurements while you're going along on the tissue of the patient, and that basically creates that nanomechanical signature of breast cancer. So in that short time that this work has been done, this is work that from Maria Blodinek in the group of Lynn of our institute. So this actually, after this short term of about 4 years, it's already available on the market as a product, as you can see here. So what we have done is we looked into whether this technology can be used as a fast detecting, diagnosing tools for illnesses all the way down to the genetic level. And that is basically in search for the unique human biology in personalized healthcare. And what we're doing is we are using arrays of cantilevers that are functionalized biochemically. And this is the operational modes that we have. So that we have a static mode where molecules are inducing a stress, and that stress basically bends the cantilevers or the sensors. And we always use references, because without that, this technology is not really appropriate. And we can also do that, it's highly reversible, and we also can do that, of course, in the dynamic mode, as in force microscopy. We know that, and there, basically, in the dynamic mode, we look at the mass or the mass changes, and here we do look at the conformational changes in the molecule. And basically what we do, the transduction, is converting biochemical processes into nanomechanical motion. We do that in a differential way. It's an instant calibration, and response time is in milliseconds. Okay? So this is some of the work we have done so far. It's mutational analysis, monitoring transcriptional activity, monitoring transcription factors, analyzing protein synthesis. We also looked to the nuclear pore, and which is part of the nuclear envelope, but time doesn't permit to do all this in all these studies. Here is some of the literature what we have published, and this is biology's requirement. So basically, if you do that using as a sensor, then no labels should be available, so no packing of molecules. The sensitivity should be 10 down to 10 picomolar. Specificity is a single point mutation, and you should be able to do that in the complete background of DNA or of RNA. And if you want to be better than the current technology, you should circumvent in one way or another the PCR amplification. So as we go along, I can show you that we actually fulfill all these requirements with our sensors. So the decoding of entire human genome is still leaving us with a puzzle. We know now what we're made of, but we don't know how we work, not yet. And there's still some of the software is missing, and one of the reasons is because there are less than 23,000 genes really are performing all function of life, and that's only 1.5% of the DNA. but 98.5% was actually attributed to kind of junk, and this non-conductive part of the DNA basically contains a lot of regulatory processes in the gene, whether genes are regulated up or down, and or are switched on or off. And this is shown here. 35,000 DNA regions are equal in mammal evolution. 150 are unique to human. And they are known as human accelerated regions. But most of them are allocated actually within this 98% of 98.5% of the non-coding and the non-coding part. So it's those regulatory processes are very important. That is only findings actually in the last 10 years since the decoding of the genome. And so people are looking into alternative splicing, but much more important is they look into the epigenome. So epigenetically changes really can turn on and off the gene activity without altering the DNA sequencing. And then, of course, this malfunctioning of these regulatory processes then trigger cancer progression. So this mutation, of course, then disrupts the biological pathway, and it leads to uncontrolled replication. So the way nature is doing this is it uses this process as methylation or histone modification. And that is done basically that it tags to these methyl groups, they tag to the DNA-based cytosine, and then it silences the gene. So that's how it really works here in this case of the cancer-fighting gene that all of us carry. And if this is switched off, it affects all its function. So basically, it silences the gene. And at the moment, nobody knows actually how to switch that gene on again. So many maladies, of course, are polygenetic, and that is having an effect epigenetically in future generations. And because it creates epi mutations, and that is really something that people are looking in at the moment. And for example, obesity is one of these illnesses that then affect future generation from actually the bottom up in the genes. So we have teamed up together with Roche Genetics, and we looked in the case of interferon alpha, whether we can see the state of the genes, whether it's switched on or off. So this worked, and I can't go in there. Time doesn't permit that today, but that is the way we are doing it. So those are single-stranded. It creates a self-assembly, a cell in a self-assembly way, and it binds with the sulfurs onto the gold part of the sensor. It basically creates a very tight monolayer, and upon hybridization, the complement is squeezing in, and it leads to a compressive or a tensile stress. And we know exactly how many molecules are actually working here, and that is be done because we have looked in and verified it with using radioactive labeling, either on the single strand or on the complement. So we know exactly how many molecules are in action, and as you could see, we use inkjet technology in order to functionalize the arrays. So this work then has been republished here, and in order to assess a patient's condition, what we do is we use conventional assay in order to create a gene pool, and then basically take the relevant biomarkers onto the array and look in whether we can see malfunctioning of these biomarkers in the regulatory process. We then started to look into human melanoma, and we do that together with the Cancer Research Institute at the University of Lausanne. So melanoma is the deadliest skin cancer, and it has a radial growth. It starts in the radial growth phase, and then it sort of goes through a vertical growth phase, and then it becomes really nasty, because it sort of contaminates the blood vessel, and it creates metastasis. And then in the circulating tumor cells, it sort of spreads throughout the body very fast. This is how the end state looks like. People who are in the state of the vertical growth phase, they have a life expectancy of about 2 months. And obviously, where I'm leading to is that diagnostic and new drug developments are on the way. So 50% of this of these patients, they actually carry a mutation in the BRAF part of the genome. And what I plotted here is a concentration dependent of that BRAF DNA, and that's between the wild type and the mutant. And you see, after injecting here and settling down, it just takes minutes in order to show the concentration dependent. And we are about sensitive to 1 nanometer deflection. And we do that in a differential way. As I just said before, without that, you can basically get no real results, because in a differential way, even if you have unspecific binding onto the device, then both are going down, and then you still have the differential way to actually get out your signal. So you actually take care of all side effects like temperature drifts and so on and so forth. This is done in the complete background of the DNA, and is done, and what we are showing here is a single point mutation. This is work from François Huber in our group, and we have taken it a step further and looked into the complete background of the total RNA. And as you can see here, also is the concentration dependence is about 100 nanogram per microliter and 20 nanogram per microliter here, both of them. And that gives us sort of a sensitivity in the picomolar region. So this is exactly what biology actually requires. On top of that, this is the point mutation in what we are showing here. And what we have taken as reference here is a simple AC/AC/AC 18-mer. And what's much more important, as a second reference, we have taken the wild type. And that is against, so these measurements are made against the wild type. And this is very important that we can actually see that there. And obviously, I mean, we always have these references, and sometimes we are passivating the lower part in order to suppress unspecific binding and things like that. And now this new work now is that we've taken a step further, that not only one mutant we can monitor. This shows actually that in this case, we have 2 different mutants where we can show that this is work. This is work in progress. So There is no labeling involved, and there is no PCR amplification here. Absolutely no PCR amplification, and I come back to this in a moment. But the question is, of course, can we also monitor the circular tumor cells and monitor those with our device? So melanoma cells express a high molecular weight melanoma-associated antigen, which this green arrows. And what we do is we functionalize the complement of the antibody and look at the interaction here. The number of CTCs is very low. So they're very scarce. So it's about 1 to 10 in the billion cell per milliliter blood. So they're very scarce to detect. And it's a worldwide effort at the moment to actually measure the CTCs in the bloodstream. And obviously, since this is a cell, we get much bigger signals, as you can see here. Again, this is the references, and the signals here are up to 6, 7, the deviation of the device, up to 6, 7 microns. Again, here we have the references, the 3 reference, and the 4 devices. They are actually showing the interaction, the antigen-antibody interaction. This is how it looks like, and you see that the cells are settling down on the device, and you have the impression that they actually stick everywhere, but that's not the case. If we purchase that, then you can see that basically only where the antigen-antibody interaction is happening, they stick to the device. And we basically have done that in a mixture of melanoma cells and lymphoblast cells. So we really not. So you can see that no lymphoblast cells are actually sticking to the device. And as you also can see, that we are close down to about 10 melanoma cells that we can monitor, 10 melanoma cells down here. So we are on the way. This is work in progress that we are able, that enables us to actually look into that we can see and detect this melanoma cells. So this is just the reproducibility of the BRAF detection. What's much more important is this. This is what Roche is using at the moment. This is the COBAS system, and what does actually tell us that we have the same sensitivity as this system. However, the big difference is that we are not using any PCR. We are not using any PCR, so we make a contribution, in particularly since Roche came out about 1.5 years ago, and with a new drug, it's known Selboraf, and this was one of the first personalized medical drug, because it only targets those people that are carrying that BRAF mutation. So since then, many more drugs actually in 1.5 years, it goes quite fast, have come on the market. And however, only after a year that showed side effects and resistance. This it just took a year in order to develop this, and now there are two new inhibitors being found, and that is also improved. Was also improved last year, FDA approval in last year, and what people have done so far, they have used them together with Selboraf. Unfortunately, they are from a different drug company. However, 300 to 400 people have have been treated with this combination, with this cocktail, and the life expectancy compared to the cell BRAF on its own was almost in the advanced state of a patient who carried these mutations or the melanoma. The life expectancy was about half a year, and the costs are enormous. So for a patient, a treatment is about $55,000. So you can see the way it develops, and we think we have a contribution, in particularly in the diagnostic part, to bring the costs down. So the rise of antimicrobial resistance is much higher than that was published around 2011, and so there is a new pattern of antibiotic resistance may leave us defenseless against wide range of bacterial infection. And the World Health Organization just came out half a year ago with the first global report of antibiotic resistance. And in particularly in MRSA, you can see that there is a high percentage of resistance against it. So that means that the standard antibiotics does not work anymore in that case. And obviously, read daily in the newspaper of outbreaks of bacteria and the resistance, and even water sources in India have been contaminated, and obviously Ebola, but that of course is a virus, but it has secondary impact, of course, that can create this. So in the hunt of this superbugs, the last sort of resort is vancomycin, the drug, and it basically binds to the mucopeptide of the cell wall, and it weakens the cell wall, and the osmotic pressure inside basically weakens the cell membrane, and that leads to cell lysis. So the bug has become resistant just by deleting one of this hydrogen bridge, just one of this hydrogen bridge. And this subtle difference can be monitored with this kind of a devices. This is work from Rachel McHenry's group, and this is just to show the specificity and sensitivity. And this is a concentration dependent, but what is important is that you can detect that in the blood serum. However, in their new data, they show they actually can do it in blood. So that's also ongoing work where we collaborate with them. So for quite some time, we looked into the microorganism detection and shrinking down the Petri dish down onto on our device. So that goes along what has been said in the talk before. To shrink down the whole mechanism is also you get faster diagnostics, and that is done here by functionalize it with Aptus, and the current methods actually show that it is 1 to 3 days to obtain useful information, 30 days, for example, in tuberculosis. Okay, and with this system, that could be shrunk down, taken down to 5 hours. 5 hours. I can't go into that particularly, but alternative phenotype methods are here. They are inexpensive but slow and read out 7 days. This side here, those are expensive, unable to distinguish living and dead bacteria. So we teamed up with Giovanni Dietler from the EPFL. So what they have done is they just have taken a AFM and a bioscope in this case, and what they have done is they injected a buffer PPS, then they injected E. coli gram-negative and bacteria in PBS. So you see these variances starting up, and if you enhance that with a LB medium enhancer, then it even grows bigger. And then if you inject antibiotics, in this case ampicillin, it just dies. So this goes just in second. All these variances, they die, and when we look at it, in all the bacteria are dead. So we teamed up with them, and we used our setup. And as you can see, is we just got exactly the same. We could validate that and verify this, and we've done exactly the same. And we got just a better statistics out of it. However, when you do this now and you inject resistant E. coli, and do the same measurement, and then inject the antibiotics, and nothing happens. So we think this is also a new way to go, and this is a new program that started actually on this next year. So let me change gear here slightly and go from the fluidic environment into the gaseous environment. So this is how the olfactory system works of a human. So in your nasal cavity in the palate, you have the millions of neurons sticking basically out. The only opening of it, it's from the brain to the outside world. And this, these olfactory neurons, they have cilia, cantilever-like sort of cilia sticking out into the nasal cavity. And upon interaction with odor and molecule and receptors, they've been transported into the axons, and it's an enzymatic amplification of 10 to the 9th. And then the first sort of readout and pattern recognition is happening in the olfactory bulb of the brain, and finally in the whole rest of the processing is done in the olfactory cortex. And when we actually started all this way back, in doing this cantilever array stuff together with Jim, we've done that, and we thought, we have actually a simplified sort of kind of nose. And you can see those cilia here. It's very interesting, actually. There are eight cilia per neuron, and we are using at that time 8 cantilevers that are basically bending. So what I show you now is not specific anymore. This is semi-specific answer, and we don't need to know the interaction between the molecules. So what we do here is we use polymers, different polymers. We can tailor-make them, and what they do is in interaction with the odor molecules, what they do is they swell, and they give a response, a different response, as you can see here, because the swelling is individual on the cantilevers, and that's what we do then is looking into a fingerprint, and we can basically do the readout as principal component analysis or artificial network. And principal component analysis is basically converting three-dimensional space into a two-dimensional projection. And what you get out here is that each measurement leads to a dot, and then many measurements, they lead to a cluster, and that is how you do the analysis. And you can see that nicely done here, shown here, with this volatile organic compounds, and we can show the difference here. For example, those are 55 measurements, and with different arrays. So
5 minutes.
okay. We have done that with way back already, and looked in, and what we could analyze is, for example, patients that are suffering from diabetes, patients that are suffering from uremia, and so on. And here you always have breast samples of healthy patients. So what you actually do, or what we are trying to do, is give the medical doctor a new device that patient can blow in to the device. I just come to that, and analyze certain illnesses. So when Genki Yoshikawa was with us, he came up with this new idea, and this is now piezoresistive. So this is a piezoresistive readout and not a laser beam detection and so on. And what came out recently, what we have done recently, is this shape. It is round now, and it contains 4 actually cantilevers. That would mean you are 4 times more sensitive. However, this depends on the thickness of the device, of these membranes, and the thinner you go, obviously, the more sensitive you will be. And this is what we have done, and it shows actually that we are 10 times more sensitive. This is the way, again, the way it is functionalized. As you can see here, with inkjet technology, we do that. And so this is how the device looks now. So here is we work together with the hospitals again in Lausanne, and so patients are blowing into that bag, and the whole electronic valves and God knows what are just in here, and it is streamed by the USB stick from the computer, so it's very mobile. You can take it anywhere, you know. And that is how it works again. You always purchase in between, and you can see the deviation and of the signals using this different kind of polymers. So we teamed up with again with the hospital in Lausanne, and we have chosen head and neck cancer to monitor head and neck cancer. And the reason for that is because this kind of cancer is not growing any metastasis. It's basically stays the way it is. And that's easier to detect. So here you go. So we have 4 patients and 4 healthy persons. This is what is called a double-blind study. So we didn't know anything. It was just given to us. We didn't know anything, and what came out is in this plot here, you can see the healthy persons, the cancer patients are here suffering from cancer, and the healthy patients are here, and here is the water, because people always said, you know, I mean, this is all part of water inflicting and all this, and as you can very well see, this is not the case. So it's not a great statistic at the moment, because this is ongoing work, but we have taken it a step further, and then we got samples that from patients they've been operated. So we have been given samples from patients in the pre-surgery state, and then after, and what you can see here, that the tendency is quite clear that after operation, they tend to go in to where the healthy people are. And in a different sort of method to look at it is this pair group method, where you look into the bifurcation of it, and there you clearly can see that the cured people or operated people, and the healthy people are in this part, and the people who are still suffering from the cancer are actually in this part. So I believe with this, we've done it also with lung cancer, and then we've done something else with, this is very new, the whole of a system has been given to Ted Terling, and what happens here is actually that in maize plant, this caterpillar here eats the leaves, so the plant doesn't like it and has a defense response built in because it releases volatile secretes, and then it attracts this wasp, and the wasp eats the caterpillar. And that could be shown very nicely with this method, and that just shows you the versatility of application that you can do with it. So actually what we're doing, we're trying to bring together the in a top-down approach and in a bottom-up approach, which is so wonderfully implemented by the natural world in self-assembly and self-organization. And I would like to finish here with the words of George Gamow that said, in the universe, as for the universe, it took just a few hours to make atoms, 100 million years of making galaxies, stars and planets, and 4.5 billion years for making a human being. And the complexity, what we see since the genome has been sequenced, and the complexity that basically builds up out of that is enormous, as we can see. And we try to understand that, of course, with various methods. So thank you for your attention.